where we look at the underrepresentation of the different populations from the point of view, for instance, of pharmacogenomics. And while previous analysis have shown the underrepresentation in genome-wide association studies, we are able to see that these patterns of underrepresentation are systemic for pretty much every genomics data set or omics data set that we get our hands on. Here, for instance, I'm just showing you data from PharmGKB, which is the leading pharmacogenomics repository for gene-drug associations. And as you can see, the European ancestry is severely overrepresented, whereas people from the Americas, and by that I don't mean European origin people, but actually native people from Latin America, they are incredibly minuscule. Their representation is minuscule. So we have reasonable representation of East Asian, and then the rest of the world is really underrepresented. So how this sort of process of Latin American happened? So with some of the data that we've analyzed, the peopling of the Americas happened around 23,000 years ago, when the divergence from East Asia, from the Bering Strait, you got the first communities coming down through the coast. And it was relatively fast, that peopling of the Americas. Around 12,000 years ago, we were able to find that they actually arrived through the Panama Isthmus into South America, and then you have three branches. You have the Amazon branch, then the coast, and then the Andes. And we have seen, I want you to remember these three sort of branches, because actually we see that consistently in further analysis. But the point I want to make here is that these three branches were mostly conditioned by the geographical characteristics of the land. So you have the coast, the mountains, and then also something that is very, very notorious about these populations is that around 30% of the total population, for instance, in Peru, they live above 2,500 meters high. And that's very, very interesting. Then when the Spanish came in 1532, there was some admixture. But as you will see in my next few slides, at that point, when the Spanish came, we had the Inca Empire, which basically extended throughout the Andean chain of mountains and then the coast. There were four main regions. And if we look at estimates of the population when the Spanish came, which happened around 1532, the effects of the Spanish conquest were absolutely devastating for the population. So from a peak of around 16 million people in the Inca population, in about 40 years, we got to about less than 2 million people. So 90% of the population died. And the truth is that actually it wasn't because, I mean, the Spanish, obviously, that I'm not trying to redeem them, but actually this is mostly explained by the fact that there were a number of pathogens that they didn't even know about, such as smallpox, flu, and tuberculosis, for which the Eurasian populations had been adapted for thousands of years, but these populations had never been exposed to that. So as soon as the Spanish came, so the actual illnesses that were affecting peoples was advancing faster than the Spanish conquest itself. So as you can see, this had tremendous effects on the population forever. And even up to today, we're still able to see the remnants of that massive, I would say, bomb that happened during those years. So if we look at how the Americas are today, so you can see that if we try to analyze where are the biggest concentration of native populations, and by native mean peoples that haven't really had mixed with Europeans or with Africans, so native people from there, you can see that the two biggest clusters, well, there are three clusters. So obviously the Inuit people in northern Canada, you have some around Mexico, but also Bolivia and Peru, they have a tremendous concentration of native populations. And while there have been huge advancements through the work that Sharon is doing in Africa, these native populations are severely, as of now, because now it's not going to happen, because that's my mission, we have a tremendous opportunity to really understand the diversity of these populations around the Andes, which, as you can see, really, they're sort of the remnants of the Inca Empire. So with that in mind, we had an initial analysis of 150 genomes. I know it's not a big number, but I just want to actually bring your attention to the fact that even though it's not a big number, we collected from 7 populations across the whole geography of Peru. And basically, they are ordered as follows. So from most indigenous, so in other words, those that have the least amount of admixture, so greater than 90%, which are the Matses and the Uros. It's quite interesting because the Matses, they live right in the middle of the Amazon jungle. You cannot see it here, but here there's the Titicaca Lake. So the Uros, they actually live on floating islands made of hyssop in the middle of the lake. And they are very, very isolated. So basically, we went in August and in September. You'll see a few pictures later on. It's quite interesting. So they are incredibly different from anything else. So, and the good news for us is that I think we're really able to access pockets of diversity that today have never been studied before. And so I've ordered the 150 genomes in 7 populations. So the most indigenous to least indigenous. So you can see the most indigenous are very isolated populations, Matses, Uros. Then you have the mountain people, Choctaws. Then you have these Moches, which is quite interesting. You don't get that, but they're quite isolated, even though they're still in the coast. And they're ordered like that. So, and the Choctaws, they are well known to be big warriors. So don't get into trouble with them. I would recommend it. So this is the breakdown of the individuals per population. So we have 7 populations there. And what I was working on was trying to understand, given that we have all this potential diversity, how much is actually novel? And what really struck me here is that around 13% of all of the variation that we observe is completely novel. It has never been seen before. And as soon as you start scratching the surface, you get new things that are, super exciting. So we perform an analysis trying to understand the high impact mutations. So mutations that basically break the gene, and then see from these high impact mutations, which are rare, by the way. So at least maximum 0.1% frequency or less or non-existent. So from these 1,200 variants, actually, the vast majority are unknown. So we have no idea of the interpretation. We have a few likely benign, a few pathogenics, but basically the vast majority of the high impact mutation that we observe, we have no information in reference databases. When I then look at the DNA count, I look at the SNP count. So this is a way of seeing how much diversity there is within the population. In other words, you can have very different genomes, but when you look at the population itself, how much variation is in that population. And here you see the Matses and the Uros. that have the least amount of diversity within themselves, meaning that they have probably been affected by a genomic bottleneck the most. And the ones that you see on the right-hand side, these are the ones from cities, so the ones that have been mixed with Europeans, and they're more sort of cosmopolitan. In the middle, you have the two ones that, one from the mountains, another one from the coast. So when we actually look at the genes that are highly mutated in these populations, but which are non-existent or rare everywhere else, we begin to see specific mutational profiles. For instance, I mentioned the Uros, the ones that live from Titicaca Islands. So these are all the genes here. And you can see, so these genes, what frequency within the population of individuals have those highly mutated pathogenic variants, and the frequency, the total frequency in the population. So when we look for specific mutational patterns within the Uros population, and then look using FIWAs, what associated phenotype they could have this, they have to do mostly with height or bone mineral density, but also autoimmune disease. So, and I'm not going to have a lot of time to show you, but I see this sort of recurrent pattern of immunity. So the genes that seem to be most affected seem to point at immunity or immune adaptation. And I have some more data to show you that. The other population, Matt says, the ones that are in the middle of the Amazon, again, the most different highly mutated rare genes in those populations have to do with immune response and then also cardiomyopathy. If I then look at from the 1,200 highly impactful variants that are most frequent in my population, and then try to look where the affected genes cluster in terms of FIWAs, in terms of the domain, I can see an overrepresentation of immunological, skeletal, and metabolic traits, a little bit on cardiovascular as well. This is the enrichment analysis. So you can see again, highly enriched those specific genes on infectious disease and immune response. And when I look at some of the annotations, for instance, in K, I can see that they relate to autoimmune diseases, genes that are related to influenza, and tuberculosis, Epstein-Barr viruses, and which happens, these two diseases, influenza and tuberculosis, were some of the two that the Spanish brought from Eurasia. So I wonder if there is any kind of correlation there, but so I get quite excited about that. So what does this mean? So we have a potential for genetic predisposition to infectious diseases, and then also an overrepresentation of genes that have to do with immune response. Then we also have around 1,000 arrays from 32 populations. So we combine the genomes with the arrays, and basically once we prune, just to avoid overrepresentation of highly related samples, we end up with around 736 samples. And now that we have a much greater array of populations as well as individuals, we want to understand more information about their ancestry, their genetic composition, their clustering, and so on. And here you can see the actual communities. Something that is quite interesting. So we have Afro-descendants, and they're basically descendants from Africa, presumably slaves that they brought hundreds of years ago. And of course, you have, again, many from the Andes, many from the Amazon jungle, and then from some big communities like Lima and other conurbations. Here you can see on the left-hand side where we took samples from. And basically, when we perform a principal component analysis, I just want to draw your attention that actually this is principal component 2 and principal component 3. Because when we have the principal component 1 and 2, basically you have the Afro-descendants. It basically separates the Africans versus the non-Africans. But then when we look a little bit into the fine-grained relations, we see very clearly the patterns of mountain, then the jungle, and then the coast, and then the Afro-descendant and the sort of ones that are quite mixed. But then the Uros, the ones that live in floating islands, they seem to have their own little cluster there. So that was quite interesting to see. Now, when we then compare them from the point of view of phylogenetics, you can see that they do have a good clustering. For instance, the ones that come from the jungle, but then not so much with the other two, the coast and the mountain. We use the Salmon Diversity Genome Project, where we basically take samples from 164 populations. And now you can see basically that the populations, I don't think you can see it, but basically in capital letters are my own populations, our own populations. And then in lowercase, there are the other ones from the Simon's Genome Diversity Projects. You can see the Afro-descendants, they go into the African cluster. But the ones that seem to be closest, you can see Eskimos, then it gets to Mongolia, Japanese. So you can really see that provenance from the Bering Strait. So it was quite interesting to see really this sort of breakdown. And then the Afro-descendants that have sort of admixture somewhere in the middle. If we then look at their actual genetic composition using a program called admixture, you can now see the different compositions. And basically what we find is that on average, people in Peru are 60% native, which is very, very high for the Americas. And when we look at the people that are more sort of isolated communities, we have had access to indigenous populations where they are less than 10% admixed. And as you can see, you can observe the ones that come from the jungle, from the Amazon jungle, and then the ones that come from the mountains. Very, very interesting patterns. The Uros, as you can see, they are incredibly different from everybody else. Again, when we look at the different clusters. We also perform a pharmacogenomics analysis. And the idea of pharmacogenomics is that by looking at the variants, specific genes that are involved in metabolization of drugs, you're going to have different appropriate dosages for the administration of that particular drug. I just want to bring your attention to this particular gene, CYP2D6. So CYP2D6 is a gene that metabolizes 25% of all prescribed drugs, according to the FDA. So 25% of all drugs are prescribed by CYP2D6. And the way this works is that if you then are able to quantify the proportion of the population that are ultra-rapid metabolizer, normal metabolizer, or poor metabolizer, you can then adjust the dosage for these patients. Something that we found quite interestingly in our research is that for sub-Saharan African, we have greater than 35% of all existing known variants in this most used gene, they are indeterminate. So we have a tremendous overrepresentation of indeterminate metabolization status for a huge chunk of the African population. And then you can see, because what happens if you have, and I think usually the risk is probably the ultra rapid metabolizers. So if you have someone, let's say you give them codeine, which is one of the drugs that CYP2 metabolizes. If you give codeine to someone, and it's a ultra rapid metabolizer, that means that they are going to metabolize it very fast. And codeine basically gets metabolized into morphine. And if you have, basically you end up having an overdose of morphine, and you could die of respiratory arrest, basically. And don't try to take it if you are pregnant either. So that information is incredibly valuable, and it tells us about specific actionable treatments that we could provide already to people without needing their phenotype. So what we did, we actually looked at all of the 150 genomes and understood or tried to analyze what pharmacogenes, so genes involved in metabolization, are affected within our population. And I want to bring your attention to F5, where basically, for all of the genomes that we analyzed, they have unfavorable response, meaning that if you, F5 is basically a sort of anticoagulant, if I remember correctly. If you give them those drugs to these people, they're not going to have the intended effect. And many of them will have actually adverse drug reactions. But something that really caught my attention is that in Peru, as it happens, it has the greatest deaths per million of severe COVID infection in the world. So they were the most affected deaths per million country in the world. And my hypothesis is that that's not just because they have a poor health system. What I did find is that gene, the F5, actually, there seems to be a link. Patients with severe coronavirus have a high frequency of that mutation in that gene. And I wonder that there is a link there. And this is kind of the sort of final observation I want to give. I can't prove it, but wherever I start scratching into the interpretation of the genomics, I always come back to the infectious diseases, to the immunity aspect. And I'm hoping that we might be able to find some kind of relation between these two, because it's kind of too coincidental. But anyway, I'll leave it there. So I'm just going to give you a few summary points. So we have found mutated genes mostly, and this is just a very rough first screening. We see an overrepresentation of genes involved in immunity and infectious diseases. And then when we combine that with anthropological measurements, with history, with COVID-19, everything seems to be pointing at the same place. And I believe that this mutation may help not just explain history, but also future outcomes in, for instance, pandemics. And also, I want to bring your attention that some people, certainly outside of Africa, this is a tremendous amount of diversity that remains to be analyzed, interpreted, and I think it's a very worthwhile endeavor. So I've just included a few pictures from my last trip to the Amazon. So these are the people we are trying to actually serve. So these are the Bora tribe. Here we are with the Uros. So as I said, they live on floating islands in the middle of Titicaca Lake. It's about a day by boat from the closest point of civilization. And then probably the highlight for me was the mother of the chief of the Bora community that, she doesn't speak Spanish or anything, but she just gave me a hug. And, that sort of stood here. And I just want to thanks for your attention. I mean, this is not just me. There's a lot of people there who actually, I'm just the person who analyzed some of that data, but these are the people really behind these amazing projects. So thank you guys for having
me. So do we have any questions? Yes.
Yeah, that's wonderful, really amazing, and also so hard to do. I don't know how you achieved that, but the few questions. The first one was about the diversity within populations. How does that compare to other populations? Because you showed it just within the ones you studied. Where does that rank? Because the axis was quite narrow, so it couldn't quite place.
How does it compare like to European?
Yeah.
Oh, right. Yeah, I haven't shown it. To be honest, I would just be speculating. So maybe next time I can give you a proper answer.
Yeah.
But obviously, I mean, we know that the Europeans are not as diverse as these populations. I actually have some data somewhere, but no, I cannot answer that question precisely. But
Then I have one other question. You say something about the susceptibility to certain diseases. Isn't the opposite probably true? Because I mean, it's one thing about the specific populations, but the other thing is about the bottleneck. So the ability to survive that pressure.
yeah,
And so, if anything, you would have thought this conveys some protection, right?
yes.
And it's obviously independent of the fact.
That's a question that I've been wrestling quite a lot, and actually the conclusion that I have currently is that you still find more heterozygosis so you should still be able to get traces of the susceptibility in the remaining populations. So they will still they will have been adapted to these new diseases, but I see a lot of heterogeneity potentially that they still will be carriers of many of those markers that confer the susceptibility, and potentially is part of the reason why you can see in current pandemics that they are more susceptible.
So when you say in terms of infection, like COVID infection in the Peru population,
Yeah.
is that also similar to other Latin American population?
Not really. But that's simply because we don't have data from other South American populations. I know there have been some analysis in Ecuador and a few in Colombia, but I've never had access or nor seen any comparison between what I'm trying to this analysis and those populations. The only thing I have is what I showed you, that there is this sort of high number of deaths per million for this population. And since we believe that this population is quite representative of the whole geography. So, and we know, for instance, I mean, I was talking with the person in Iquitos. So the health responsible of Iquitos here, they were notorious for the number of deaths that they had. I mean, it was just unbelievable around this area, which happens to be some of the ones that we've studied. But I can't give conclusive evidence. I can only provide conjectures and hypotheses.
Yeah, I think in order to rule out the maybe lack of good health system as the cause for the high deaths. So we need to check what is happening with other close by population.
Yes. As I said, we only have these populations. And then the analysis that we got, the statistics, they put Peru all together in just so there's no granularity between the different communities. Right. Because the statistics, we kind of assume that the sort of statistics that we have for World Health Organization you can get from Europe. is very different from the type of statistics that you can get from here. That's another challenge as well.
Who is on? Oh, yes.
Yeah, thank you for your presentation. And it shows great work that you are doing there and how much acceptance you have gained amongst the people, which is very interesting with the pictures you've shown us. But I just wanted to find out in terms of communicating back results to them, the outcome of this. So how do you sort of engage with them to interpret this to them in a way that they will understand, they would appreciate what you are doing?
This is a fascinating question, and to be honest with you, that's the other half of the work. So I'm showing you the sort of nice research, but a lot of what we are trying to do is the engagement aspects and the social connection. And yes, it's a tremendous challenge. So that's what we did in the trips that we've done before. And basically, it's all about talking to them, understanding what they care, and then obviously trying to explain to them DNA when they live in the middle of the jungle. But I was surprised because we start talking to them and then we say, do you have some relatives or people that get sick and they seem to sort of be in the family some diseases? Oh yeah, they have this sort of hemorrhage, and we can only palliate. We don't have any cure or anything. Okay. So would you be happy if we maybe have a little look and see if we can perhaps understand it a little bit better from your point of view? So that it was kind of that kind of conversation, and I was surprised by how they were so eager, like please. Like, no, we have to do it the right way. We have to have the permissions, we have to have the appropriate protocols. So in terms of returning results, that's absolutely essential, and it's kind of part of the social contract that we have with them. Unfortunately, we cannot cure them. We don't have specific treatments to give them, but that's precisely what we are trying to work on right now. So what do we feed them back with? And we will show them things to do, for instance, with high altitudes. As I said earlier, 30% of the population lives in a high, and we have seen potentially some cardiovascular disease mutations and susceptibilities. We also, as I said earlier, we also see things to do with immunity. And so we can't give them a genetic report, but what we are trying to do is to have like very, very sort of simple pictures and booklets where we tell them, about why this is important and how this is useful. Maybe not for them now, but for their descendants, it'll be very useful. I'm not saying that this is the right type of intervention, but at least we do impose upon ourselves that we're not just going to go back and not give them anything. We want to give them something, which even if it doesn't help them now, that at least they see that some return for their investment. And then the other thing I feel very strongly about is that the sample sequencing, the sample collection, everything should be done in Peru. Because at the moment, basically what everyone does, they get the samples, they send them somewhere else, the samples get analyzed, somebody else also benefits, and the skills, the capacity escapes this country. And I think that's finished now. And so what we want to make sure is that the samples, the training, the capability is in Peru. So, and that's probably the best way we can help and we can benefit the community, even if it's not there. But if it just can stay in Lima, that already will make a difference, I hope.
Thanks. Yes.
The data is accessible via EGA and via sort of controlled access. It's on EGA.
Okay, so Claudia, I want to ask you, this is just 150 sample. So what's
Actually 1,000, but
8,000? Can you what's your like a next big stuff that you want to do in summary of what you want to do next after all this?
why are you asking me? You know it.
No, just okay. Tell us.
I want to, well, my dream, and anyone that hears our story, we want to develop a proper biobank. So the next step is go to 50,000 samples, and we are in the process of applying for a very cheap project of $50 million. And there are some people, there's one organization that is interested. I'm not sure if it will happen. We are working on it. But the idea, so currently we're just based in Peru, but my dream is that, you know very well what's happening in Africa, right? You have the Wellcome Trust, now you have this Genomics Institute that serves Africa.
Yeah.
What that means is that there is a Pan-Africa organization that is able to capture international funding. That funding, and this is the beauty of H3 Africa.
Yeah.
You have a sort of auction process whereby local scientists can apply for funding that is for local projects, for local researchers. And that organization doesn't exist in Latin America. And that means that there's no coordination or federation, and the impact is limited to the national initiatives, which there have been. But that, to my mind, is limiting the potential impacts in terms of scale if you were able to actually integrate the data from many different countries, like is happening in Africa. So my wish and my vision, my mission, whatever it takes, I want to have something that is maybe not the same, but equivalent and interoperable. Because would it be nice that actually we can compare African genomes with Latin American genomes? Because the needs and the diseases are very different from the north. We are talking about the global south.
Yeah.
The global south, these underserved populations have very specific needs that are not necessarily what we have here in the West or in industrialized nations. And so we've been talking about an Afro-Latin genome diversity biobank. We've been talking to people, so that's also something that is our ambition.
Yeah, I mean, I don't know, it's a difficult question. I think it's also, much of what's happening in Africa, much of what's happened here, certainly in terms of sequencing, is driven by commercial interests. And so much of even of the kind of genomic commercial sector is not necessarily driven by people's interest in health, but in interest in their relatives. So, I mean, I wonder whether that's also a kind of, there's no human exceptionality. That may be exactly the same in some of those communities, to understand who in terms of populations and people is close to you or more distant. And so that in itself is not new knowledge, right? And new understanding and interest. And I mean, I just wish there could be something like 23andMe, which is a great idea if it was an academically and locally driven initiative rather than, as we say, the helicopter or, overeducated, healthy, who kind of go into these and then benefit certain groups. But the concept of that is very
Claudia, I'm actually quite hopeful because, I mean, it's not, things have changed dramatically in the past, probably since George Floyd. I mean, like, there's I mean, you see everywhere that there's massive. People are aware that this is so critical. It has to change, and what we are trying to do in terms of leveraging the commercial interests is that it's fine, actually, as long as X% of whatever you discover for new drugs,
Yeah.
we get it back for the people. And this is something that is happening, for instance, in Oceania. I've also seen it in Canada. And so, and the pharmaceutical companies and whatever is interested in this, you know, they are not that averse anymore to that sort of capacity building as sort of part of the deal. So you can have the data, but you know we'll have the data as well, and you support our infrastructure, our research, and whatever you manage to do in terms of profit, you share it with us as well, and the money stays here for the economy, for them. I mean, I think that I'm quite sort of hopeful because I mean, even in these few years, I've noticed change. Have you not?
Yeah.
Like people are a lot more aware that, you know, we can't have precision medicine if, you know, if it's only serving like 20% of the global population. I mean, it doesn't make any sense.
Yeah.
So I think right now, honestly, is the time, and it's incredibly exciting to be able to work in what we are doing. I think we're going to have jobs
Yeah.
for the next 20 years, aren't we?
Sure. Okay, I think that's everything. Please let's appreciate Mane once again. Thank you very much for coming. Thank you. Okay, great. Oh, is there any question from you? No, okay. Hey, bro. Thank you very much. That was good.